Hello everyone. In this video, I want to discuss about gastrointestinal GI microbiome imbalance and the changes in the immune system responses to the microbiome which leads to inflammatory bowel disease IBD. First, we examine the genes and cellular interaction which involved in the development of IBD. Then we statically analyze the changes of bacterial diversity in phylum and general level, also the effect of the environmental variables such as treatment, mesalamine, on changes in the intestinal microbiome diversity. Irritable bowel disease (IBD) refers to uncontrolled inflammation in the intestines. Such inflammation is associated with abdominal pain, diarrhea, bleeding, and loss of appetite. As we can see in this picture, there are two types of IBD. The first one is ulcerative colitis UC, which found only in the colon, and the other is Crohn's disease CD, can be found anywhere in the gastrointestinal tract. IBD occur for various reasons such as genetic allergies, stress, and so many other factors. Also, it has been increased with industrialization foods, fatty foods, and fast food, and they took the place of fiber-rich sources in human diet. Gastrointestinal GI microbiome is the gut microbiome is a combination of fungi, bacteria, and even certain viruses that can be varied depending on age, sex, and disease condition. In this picture, we can see with the increasing the age and environmental factor, the composition of microbiome changes. The human gut microbiome is made of four phyla of bacteria, two phyla Firmicutes and Bacteroidetes make up the most of the bacteria, and the two of other phyla is Proteobacteria and Actinobacteria, and they function to regulate the immune system and development of intestinal tissue. Mesalamine is the most common medication for IBD, which can prevent the inflammation, and also fecal microbial transplantation (FMT) is a new treatment which is under investigation. There are too many genes involved in IBD. The first one is NOD2 gene, which encodes the intercellular recognition receptor, and also the ATG16L1 involved in the process of autophagy, and the gene is involved with mucin synthesis and act as a physical layer and protective barrier in the intestinal epithelium. After selecting the cohort, we place them in two categories, healthy and patients. Some patients were then treated with mesalamine, so biopsy and stool samples were collected, and then DNA was extracted from them. They were amplified by PCR for using 16S sequence. Then these fragments were sequenced and used to bioinformatic method to estimate intestinal diversity. This table shows the detail of the hospitalized patients such as number, gender, and age by using the colonoscopic examination. The disease and its type of the UC or CD were diagnosed. DNA contents were extracted by using the QIA-AMP kit and were used to separate DNA fragments. Then the relative abundance of each genus of bacteria was calculated by Shannon Diversity Index. Studies have shown that smoking reduces the diversity of intestinal symbionts. By increasing age, we see the decrease in phylum microbiome, and antibiotics generally reduce the frequency of members of genus gram-positive bacteria, but this study shows that the abundance of bacteria and their diversity are significantly reduced in IBD conditions before and after referred to mesalamine treatment. Under certain conditions, E. coli has shown pathogenic and invasive properties to intestinal mucosa. E. coli member cause cell cycle defects, DNA damage, induce an immune response, and inflammation. In addition, Bacteroidetes and proteobacteria are significantly reduced in the treatment of mesalamine, which improve inflammatory condition. So alternation in the microbiome are more functional than ecological. The main function of mesalamine is to prevent inflammation by preventing the expression of immune factor. The cascade of inflammatory reaction lead to production of large amount of oxygen-free radicals and oxidative stress. The image shows new LBP treatment strategy and obstacle ahead. Thank you for your attention.